Okay, we have now arrived at a second series of lectures, which I have collectively referred to as the oral virome. And these are the things we're going to talk about, taking them in order. And the first item is summarized on the next slide. Okay, so the question here is how to All right, let's move on to the blood-borne pathogens, represented by members of the first four groups of viruses listed here, the four on the upper left, if you like. These pathogens are still around, and they still pose challenges for dental health care workers, which is why you wear gloves and face masks, even when it's not Halloween. Face masks, of course, also serve to protect you from viruses that are normally spread in aerosols. More of this later. That said, the occupational risk of contracting one of the blood-borne viruses in the clinic is actually pretty low, provided that standard precautionary measures are observed. All right, let's consider these viruses in the order given. So beginning with the retroviruses, there are a few examples here. I'll tell you what they are. You don't have to know that. But, the first example shown here is mouse memory tumor virus. You can see these viruses acquire an envelope by budding through the plasma membrane. The next virus is SIV, which stands for simian, meaning monkey, immunodeficiency virus. So it's closely related to HIV, which is the final picture in the form of a diagram, which is also presented in the next slide following this one. So continuing with the life cycle of this virus, so when after the virus infects a cell, a cDNA copy, and it'll be double-stranded DNA by the time this process is finished, is made using the enzyme reverse transcriptase. That cDNA copy is permanently integrated into chromosomal DNA of susceptible host cells, and the integrated DNA is then transcribed to make RNA, messenger RNA, and genomic RNA. Progeny core particles are produced, and then these exit the cell by budding through the plasma membrane. HIV, as you've no doubt heard, is immunosuppressive, in part because it targets helper T cells, also known as CD4 plus cells. CD4 being one of two receptors for this virus. When the CD4 plus cell count goes below 200 cells per milliliter, then an HIV infection will progress to full-blown AIDS unless treated. The most common oral manifestations associated with AIDS are illustrated here. On the left we have hairy leukoplakia. Man, that's a horrible term. It's caused by Epstein-Barr virus, which is a type of herpes virus. And on the right we have thrush, which is also called candidiasis. It's caused by a yeast, Candida albicans. If your patient displays either of these symptoms, he or she needs to be tested for HIV as soon as possible. An increased incidence of oral ulcers and Kaposi's sarcoma lesions are also associated with HIV. Infection. Infection is also associated with increased levels of herpes virus in periodontal pockets. So whether herpes virus itself is a direct cause or a consequence of periodontal disease is still somewhat uncertain. I'll say more about that a bit later. HIV infection will affect the function of the salivary glands, right, which secrete, among other things, a molecule referred to as histatin 5. Histatin is Histatin, along with some other potent antifungal peptides. And it's for that reason that HIV or AIDS patients often display candidiasis. All right, let's now turn our attention to the blood-borne hepatitis viruses, beginning with the most contagious one, namely hepatitis B, which is described on this particular slide. It's the most contagious for the simple reason that there's much more of it in circulation than is the case for any of the other viruses. For example, quantities of hep B often exceed 10 to the 10th per milliliter in blood, whereas hep C is rarely above 10 to the 6th per milliliter. And so for this reason, the risk of needle stick transmission is much higher, 20% for hep B versus 2% for hep C. Now, all of this said, you will all, presumably, have already been vaccinated against hep B using Twinrix, which also protects against hepatitis A, which is a totally different kind of virus, and also protects against hep D, which is bloodborne, and has a high mortality rate, up to 20%. It's largely associated with intravenous drug use. But hep D is what is called a satellite virus, meaning it cannot replicate unless another virus, another specific virus is present. And that virus is hep B, so if you've been vaccinated against hep B, you're not going to get hep D either. Right, we'll move on to the flaviviruses, of which there is only one, hep C, which is blood-borne. Hep C belongs to a rather nasty group of viruses, several of which are moving northward in spite of the climate change deniers. Within this group, there is an effective vaccine at this point only for yellow fever, although dengue vaccines are being developed, and one for Zika could appear at any time. There's no vaccine for hep C, but there are a number of very good antiviral compounds, one of which, brand name Harvoni, Harvoni, is said to have a cure rate of 94 to 99% in about 8 to 12 weeks. It could be argued that most of the flaviviruses are nasty because they're not well adapted to humans. With the exception of hep C, they are all, in fact, insect viruses, and so are particularly well adapted to insects, particularly mosquitoes. All right, let's consider some other virus families now. I guess at this point in time, with the pandemic going on, the type of virus most concerning to healthcare workers of all kinds would be the virus that causes COVID-19. But aside from that, and I'm going to be talking about that virus at the end, but aside from that, it's viruses in the herpes virus family that are perhaps most relevant to dental healthcare workers for a number of reasons. Oral lesions of viral origin will most likely have been caused by herpes simplex virus. And secondly, 3 herpes viruses, namely Epstein-Barr virus, herpes simplex virus type 1, and human cytomegalovirus, have been linked to periodontitis. As dental practitioners, you wear gloves primarily because of herpetic whitlow, which is shown in the next slide. Due to herpes simplex virus infection caused by bite wounds. The herpes viruses are among the most complex affecting humans. Their genomes encode a minimum of 150 different proteins, of which less than half are required for replication in vitro, suggesting that a large number of herpes virus specific proteins are likely involved in manipulating their host, or perhaps in surviving the environment outside of the host. Anyway, it's no surprise to learn that these viruses can manipulate host cells to their own benefit. And in point of fact, the herpes viruses are extremely well adapted to their hosts, rarely causing severe disease. As we've seen before, the herpes virus nucleocapsid gains entry into the cytoplasm via membrane fusion, followed by transport of the nucleocapsid to the nucleus and uncoating at nuclear pores. This initiates a primary infection at the initial site of entry, which would be the oral mucosa in the case of HSV1 and HSV2. All herpes viruses set up a permanent, latent infection in the host organism. In the case of HSV1, this occurs in a nearby peripheral ganglion. What happens is that all virus-specific transcription is shut down, which allows the virus to evade immune surveillance. From time to time, various stressors, like stress, will trigger reactivation of the virus, such that some replication will occur in the infected neurons. Progeny nucleocapsids will then travel along axons back to the original site of entry, and then guess what? You develop a cold sore. Latency allows herpes viruses to permanently colonize the host organism in a relatively friendly way, with periodic reactivation leading to transmission to additional hosts. I mentioned earlier, in the context of polymerase chain reaction diagnostics, a possible link between certain herpes viruses, namely Epstein-Barr virus and human cytomegalovirus, and periodontitis. Now, while there's a very strong correlation between herpes virus prevalence and severe periodontitis, this doesn't necessarily prove causation. It could be that specific bacterial species cause periodontitis, which could then be exacerbated by the presence of one or more herpes viruses. Definitive proof of a causal relationship between herpes viruses and periodontitis likely awaits the development of an effective vaccine against these herpes viruses. So that said, with regard to a link between a herpes virus and periodontitis, this particular paper demonstrates that there very likely is, in this particular instance at least, a causal relationship. Simply because antibiotics did not have any effect on the disease, whereas an antiviral did. The antiviral therapy, use of acyclovir, is therefore warranted in some cases and may clear up a nasty lesion. Here is acyclovir, and I should point out here that there are two spellings of the word. One of them begins ACI, as you see here. Another common spelling is ACY. Whatever, either one is fine. Acyclovir is a guanine analog, which becomes phosphorylated by a virus enzyme and then by a host enzyme. The triphosphate then becomes incorporated into replicating DNA, pairing with cytosine residues. This inhibits further DNA synthesis, which then halts. All right, we now come to the picornaviruses, which are described on this particular slide. One of the reasons dental healthcare workers wear face masks is to protect against aerosol-borne viruses such as the rhinoviruses, which are a common cause of the common cold. As for hepatitis A, which is another picornavirus, it's not much of a problem in North America, but is prevalent in Latin America. So, the only picornaviruses causing oral lesions are the Coxsackies and a few of the enteroviruses. Coxsackie A16 is the usual cause of hand, foot, and mouth disease, shown in the lower photograph. But enterovirus 71 infection can cause the same syndrome. Enterovirus 71 has also been associated with severe neurological complications in children under 5. Anyway, often there will be a sore mouth for a few days before ulcers and blisters appear in the oral cavity, so infected young children may refuse to eat. Vaccines against enterovirus 71 have been developed primarily in China. Of local interest, as you can see from this slide, while outbreaks of hand, foot, and mouth disease are not common amongst adults, it can happen, as we see here. Usually, outbreaks of hand, foot, and mouth disease are linked to daycare centers. Viruses causing hand, foot, and mouth disease are enteric pathogens, which means that their main site of replication is intestinal. which means that progeny virions will be present in feces. Transmission is therefore primarily hand to mouth. Obviously, the virus can also be picked up by contact with blisters and contaminated saliva. All of the picornaviruses set up an acute, self-limiting infection. We now come to the human papillomaviruses, for which there are some rather alarming numbers, as given in the next slide. That said, over 90% of infections are cleared within a couple of years. It's not entirely clear why this is the case. Let me move on to the next slide, actually. Yeah, I wanted to talk about the numbers here. As you can see, the proportion of throat cancers caused by human papillomavirus is increasing. It's not entirely clear why this is the case, but given the length of time between an initial infection and the appearance of a tumor, some have speculated that the numbers might reflect an increased level of sexual activity beginning in the 1960s and involving multiple partners. If this makes sense, then the incidence of oral carcinoma will probably not decline for another decade or so. With regard to the oncogenic HPVs, namely HPV 16 and 18, there are others, but these are the main culprits, which are commonly thought to be transmitted primarily by sexual activity. You should keep in mind a couple of things. First, it's easy to find HPV on the fingertips of individuals with genital warts. And two, HPV can remain viable at room temperature for several days. So hand-to-mouth transmission is likely more common than is generally assumed. And you should know, again, from a previous slide, that a couple of really good vaccines exist, and all children should be vaccinated, of both sexes. The next slide we're going to ignore. But there is something else I wanted to say about HPV 16. And this is probably true of 18 as well. Cancers associated with these serotypes are more readily cured using radiation therapy than oral cancers not caused by HPV. It turns out that the HPV E7 oncoprotein, an oncoprotein is a protein that promotes the development of tumors. Anyway, this protein promotes an error-prone DNA repair pathway. So that means a whole bunch of DNA breaks. This makes the tumors more susceptible than others, than non-viral induced tumors might be, to radiation therapy. And in fact, tumors caused by HPV 16 and 18 in the oral cavity are fairly easily treated if they're caught early enough. All right, we come to the paramyxovirus family. We're not going to spend much time on this. I only bring it up because there is an oral manifestation comprised of clustered white spots on the buccal mucosa, resembling somewhat grains of salt on a damp background. These are referred to as complex spots, and they represent an early manifestation of measles infection appearing 2 or 3 days prior to an actual measles type rash. So they are of some importance because early diagnostics can lead to isolation and should lead to isolation of the patient, thus inhibiting spread of the infection. Measles is a potentially serious disease which can lead to fever, deafness, brain swelling, and even death. The virus is quite hardy and can survive in the air for up to 2 hours after being released by a sneeze or a cough. There is, of course, a very good vaccine, and so the disease is entirely preventable. As I've implied, measles is an extremely contagious pathogen in which herd immunity has been diminished worldwide because of the nonsense propagated by anti-vaxxers. I hope you guys aren't in that group. All right, I think I said before, ignore slide 23. Now here we are with the togaviruses. Only one virus, rubella, is of interest to the dental community. And I bring it up because it can be transmitted congenitally. And when that happens, there can be severe repercussions vis-a-vis oral health. This is because congenitally infected children may have poor manual dexterity and so may do a poor job of brushing their teeth, in which case you can expect to see a lot of cavities. Last but not least, particularly these days, we reach the coronaviruses. In point of fact, dental health professionals should be more worried about this virus than any of the others I've talked about so far. There are, I think, about 7 of them that infect humans, with 4 of those responsible for about a third of common colds. The other common colds are caused by rhinoviruses, of course. The nasty ones are referred to as MERS, MERS, which stands for Middle East Respiratory Syndrome, or SARS, which stands for Severe Acute Respiratory Syndrome. The most recent pest, SARS-CoV-2, causes a disease called COVID-19, so-called because it was discovered in late 2019. While apparently most infections are not severe, the fatality rate is nonetheless currently estimated to be somewhere between 2 and 4%, as compared to, say, 0.1% for the seasonal flu. It's dangerous because it's very easily transmitted from person to person. And the results of that we are currently experiencing. SARS-CoV-2 most likely emerged from bats. My advice would be that you keep away from patients, except for dire emergencies, in which case you need to be wearing an N95 carbon respiratory mask. Until the pandemic is over, all patients displaying any symptoms, including loss of smell, which is a symptom in many cases, should not be seeing their dentist. Okay, we come to the last item on my list, which I've already talked about previously, namely the use of bacteriophage. In this case, against bacteria in the oral cavity. So, since many, if not most, problems in the oral cavity originate from bacterial activity, and since many bacterial pathogens are now resistant to antibiotics, why not investigate bacteriophage? Some of which, just to remind you of what they look like, are illustrated in the next slide. And the next two slides are simply reminders of what phage therapy can accomplish. Following this, we have a micrograph of Enterococcus faecalis. So this is Enterococcus faecalis. It's actually a picture that I took. Many years ago. Anyway, it's a big problem for the oral surgeon because it can cause persistent root canal infections, and it's really difficult to get rid of, and most strains are resistant to commonly used antibiotics. This bug is also a well-known cause of septicemia and can thereby contribute to endocarditis and rheumatoid arthritis. So, in the past few years, some labs have been focused on isolating bacteriophage specific for this organism, and there has been considerable success in this endeavor, as the next few slides will show. I've been throwing them in just for reference purposes. It's just an area of dentistry that I think will, down the road, acquire more and more significance. And so I just want you to be aware that this kind of progress is occurring and is in development. So just something to watch out for. And as I said in a previous lecture, I can see the day coming when you will gargle with bacterial viruses rather than Listerine. Okay, it's been nice talking to you guys. I wish it had been face-to-face, but regrettably we haven't been able to do that. Take care.